Is Dan Stamer. Dan got his Bachelor of Science and PhD at the University of Arizona. He was a professor of ophthalmology and pharmacology at the University of Arizona. In 2011, he moved to Duke, and he's now the Joseph Wadsworth Professor of Ophthalmology. Got that right? Good. His interests are cellular tissue and organ culture model systems, and also with the mouse eye, looking at conventional outflow. His talk today is pharmacology and physiology, including model systems, of aqueous humor outflow. Dan?
Thanks, John, and thanks everybody for coming and participating in this course. And hopefully now that everybody's caffeinated, you'll stay in tune for the talk. So I'm going to try to, these are my disclosures, and like Arthur, this is the first time they all apply to this talk. I'm going to try to overlay on with what Ernst just presented. And specifically, in addition to outflow, I'm going to talk about aqueous humor production and uveal or unconventional outflow, and specifically related to the Goldman equation and how the different parameters are affected. Going to be looking at most of the talk conventional outflow, and going to be ending with ways to impact outflow resistance, and really what sort of drugs can we envision to target this tissue, and which ones are actually in development currently. And then if there's time, and I hope there is, depending upon questions, talk about model systems. So this is a primer for a lot of people in the audience, and really want to, like Ernst, point out some of the advantages and disadvantages of the different models that are out there for you guys to consider, especially if you're studying conventional outflow, which is my area of research. So this has been described very well by Ernst, but there's an equation called the Goldman equation that it sort of balances the different forces, the hydrodynamic forces in the eye that give us IOP. And of course, the fluid is produced in the ciliary body, or by the ciliary processes, and it exits via two pathways: the unconventional pathway that he described, as well as the conventional pathway. And just remember that the conventional pathway or outflow facility is the inverse of the resistance. So it's that is the resistor in the eye. And then there's this back pressure on the system, which is created by the episcleral venous pressure. And so, Arthur, this is the slide that's most commonly abused. But the point of the slide is that if you do lower pressure, the vision is preserved, and it's a dose-response relationship. So pressure is important, and there's been many studies since this study looking at low-tension glaucoma, people with normal pressures, showing if you lower pressure in those people's eyes that they also do better. But the big problem is that we can't get it down enough, particularly medically. That study was more surgical application, so we really want to look at medically. And so we have very good ways medically of decreases inflow, and actually some surgical ways of doing that. I'm just going to focus today just on the pharmacology. We can increase uveoscleral outflow. We got a very good drug to do that. And we've got some ways, mostly surgical now, to increase outflow. But we've got a couple drugs in development that I will describe at the end of the talk that are targeting conventional outflow. So this was a meta-analysis that was done recently, and I liked it because it looked at the efficacy of all the drugs that are being prescribed. And to no surprise, the prostaglandin drugs were in first, and this is the drug class that targets unconventional outflow. And then we're looking at other drugs like brimonidine and timolol, the beta blockers, the adrenergic agents, and then the carbonic anhydrase inhibitors are at the end. But all of these drugs, do one thing, and that's lower pressure. So what I'm going to start with is looking at the drugs that and the physiology of how aqueous humor is produced. And so Ernst briefly mentioned that it's actively secreted. And the reason is because you've got these forces, and this is just a schematic of a ciliary process, and you've got all this blood that's inside the ciliary process. So you have this hydrostatic pressure from the capillaries pressing outward. You have an oncotic pressure that's wanting to drive fluid back into the ciliary process. Then you have interocular pressure in the eye also forcing. So these things sort of basically cancel out. So in order to get fluid out of the ciliary process, you have to secrete it. So how do we do that? Well, just like any other epithelium, you need to move salt from one side to the other. But this isn't any epithelium, it's an epithelial bilayer. And what Ernst didn't mention is that it's unlike any other tissue in the body where you have an apical interaction with gap junctions. And what you need to do is, the way this works is that fluid from the stroma comes in to the pigmented epithelium. It's transported in the pigmented epithelium by a series of transporters and antiporters to move mostly bicarbonate into the pigmented epithelium, and then carbonic anhydrase is cleaved, and that drives the salt, the sodium and the chloride, into the epithelium. And then there's these series of gap junctions that connect on the apical side between the two cells. It just freely diffuses into the non-pigmented epithelium, and then it's transported out primarily by the sodium potassium ATPase into the posterior chamber of the eye. And that's where you get the sodium chloride. And the two drug classes that we use to target this, of course, carbonic anhydrase inhibitors. If you disable that carbonic anhydrase enzyme in the pigmented epithelium, you sort of arrest that initial step. Whereas the adrenergics, they do a couple things. One of them is to affect some of the transporters in the pigmented epithelium, the non-pigmented epithelium, but also they also modify the blood flow to the tissue as well. So aqueous humor. So it's pretty much isotonic with serum in the blood, and the plasma in the blood. The exception is that it concentrates by, I think it's about 300-fold ascorbate, vitamin C, which is a reducing agent that you can imagine in the aqueous humor where you have a lot of UV light coming into the eye, that you want to have something as an antioxidant. The other thing that you don't have is protein. So a lot of the protein is not allowed to come in, and you can imagine that also light coming into the eye, you don't want to have protein in the way of that light. So what are the functions of aqueous humor? Hydromechanical, that was mentioned earlier by Ernst. It inflates the globe and keeps all the optical structures in their proper place. And so you can imagine evolutionarily that ocular hypertension is going to be favored over hypotony, just because you want to keep those structures in place. And so you're going to have sort of a skew to the curve to that side. And unfortunately, some of those people cannot tolerate that elevated pressure. Transport, right, it's the eye's circulation system. It's giving nutrients and removing waste from tissues like the crystalline lens, which doesn't have a blood system, the cornea, which doesn't have a blood supply, and the trabecular meshwork, which is also avascular. So you need aqueous humor to move these things around. And it also it provides, remember, this is an extension, this is an outpouching of the brain that's coming into, that's facing the outside world. So you need a nice protected environment for those tissues. And it has its own blood-brain barrier. It has the blood-retinal barrier and the blood-aqueous barrier, which I'm going to talk more about in detail. And it provides an immune privilege, as you can imagine, because of that. And it also, the aqueous humor provides both cellular and humoral immune responses under adverse conditions, such as inflammation and infection. What we don't know, and there's some evidence back and forth, is if there are signaling molecules that are carried from the inflow tract to the outflow tract. My guess is there are, and those are important. But this is also an understudied area that's open for you guys. So unconventional outflow. Ernst mentioned it briefly. Aqueous humor enters at the iris root between the ciliary muscle bundles. Then it goes into the choroid, where it exits via two pathways, the vortex veins, which is often called the uveo-vortex pathway, or across the sclera and the episclera, which is called the uveoscleral. So there was a wonderful review that came out by Mark Johnson and Daryl Overby, looking at just describing the uveoscleral pathway. And they preferred to use the unconventional name instead of uveoscleral or uveovortex because it's encompassing of these two pathways. So it is pressure dependent in the sense that fluid has to move in line with the pressure gradient, but it's insensitive to IOP changes in terms of the amount of fluid that exits the eye. It decreases with age, often in association with presbyopia. And there's a couple ways to measure it. The best way, and the way I'm going to show, is by tracer because you're actually directly measuring it. The other way is by indirect, which means you use the Goldman equation, you do measurements for all the other components, IOP and outflow facility and inflow, and then what you do is then you do math to figure out what the uveosclera should have been. So it's everything, it's sort of the bucket that nothing else fits into. The problem is there's errors in each one of those measurements, which means, you know, by the time you get to predicting what uveosclera is, sometimes it's negative. And so that's usually not good. So it's better to look at the tracers if you can. Obviously in humans that's not practical, at least not these days until we get some imaging that we can do that. So how much of flow is uveoscleral? It depends on what species you are. Human, and they can keep in mind these are older eyes. So these are probably 70 and 80-year-olds that were looked at. It's very little. 10% is usually a number thrown around in older eyes. It can be up to 40% in younger eyes. And I think having an active ciliary muscle plays a part in that. And that's opposed to in monkeys, which tend to have a higher percentage of outflow going through this unconventional pathway. And I forgot to mention that McLaren was also part of that. This is the reviews right here that just came out. So Ernst showed this picture of the ciliary muscle. And it was a wonderful piece of work because it showed a couple things. It shows on the top when you arrest the muscle with atropine. So this is a cholinergic antagonist. So the muscle is disabled and totally relaxed. You can see the muscle pooch up, and you can see they used I think is it called Hendrix stain, Ernst, that can trace the spaces between the ciliary muscle, and you can see on the top panel that those spaces are open. When the muscle shortens, as you do when you contract it with pilocarpine, those disappear. And why is this important? Because if you put a tracer in the eye and you take a look at the tracer going through these two pathways in the same drug conditions, you've totally shut down uveoscleral outflow by contracting that muscle. And so it's sort of a neat way to look at its function in experiments, but I think it also gets to the point of where that fluid's going. And even more interesting, this is also work that Elke and Ernst did, showing a ciliary muscle on the left that hadn't been treated, and this is the monkey, and then one that's been treated with prostaglandin F2 alpha. And so these are the prostaglandin drugs that are the first-line drugs like latanoprost for treating glaucoma currently. And what you notice after, I believe it was 3 months treatment, the spaces in between, and this has no cholinergics involved in this is just after treatment, have opened up. So you've opened up those spaces. The extracellular matrix between the ciliary muscle bundles are gone, and so fluid is flowing through that pathway. And if you take a look at actually the enzymes that are responsible for clearing all those pathways, they've increased, they've doubled in their expression in that tissue. And then there's some great pharmacology that Paul Kaufman did, where if you, Interocular pressure is here, and we're just looking at time over here. And this, if you, in the presence of pilocarpine, in the presence of contracting the muscle, you've ablated this IOP lowering effect of the prostaglandin. And then if you include, if you basically include atropine in the equation, you regain it again. So you relax the muscle, the fluid goes through there. So you're showing the mechanism of action of this prostaglandin drug. Very nicely done study. So are there any questions to this point on either inflow or uveoscleral outflow? I'm going to spend most of the time on conventional outflow just because that's, I think, the coolest tissue, but okay, I'll go ahead and move on. Right, so yeah, you're basically, yeah, sorry. So the question is, and I'm not a clinician, so I can't speak, but I'm a pharmacologist, so I can talk to that point. Does it make sense clinically not to put a patient on a prostaglandin and a pilocarpine at the same time? I guess it depends what you're doing with that patient, but yeah, I would agree they're counteracting each other. Pilocarpine isn't commonly used nowadays because of the side effects that Arthur pointed out, but it's a great research tool. So. And you probably can speak better to the clinical reasons to use pilocarpine than I can. So, so conventional outflow. So we've known for quite some time that. So this is patients. So we're looking at pressure and facility of outflow. And this is Dr. Grant. These were untreated patients, and basically just looked at their pressure. And there's a way which if we have time to get to, that you can measure outflow in human patients. And you can see right away that I mean, it's pretty darn striking that there's two groups that segregate right away. You got sort of this group that is lower resistance, but and low pressure, and then higher resistance and a high pressure. And it turns out this group is the glaucomatous and this group isn't. So they line right up. This is saying that high resistance to conventional outflow responsible for this IOP. So this has been known for quite some time, since the 50s. And this is the tissue that Ernst talked about, fluid driving, pressure driving fluid across the tissue. I just want to point out that, and he didn't cover too much, but so you have the circular Schlemm's canal that fluid's entering. You have all this stuff that's beyond Schlemm's canal that we'll get back to. This is the distal part of the outflow pathway. This is all part of the conventional outflow pathway as well. And one other thing that Ernst didn't mention is around the circumference of the tissue. So here's Schlemm's canal, here's this distal pathway, here are these collector channels that appear every so often. If you take a look and you perfuse tracer into that tissue, there's hot spots. So, and particularly in older eyes, you see areas that are functional and areas that are non-functional. And at least in this moment, we don't know what the lifetime of that tissue is and whether these are moving fluid at a later time and they're switching off. We just don't know. This is another open question that needs to be answered and areas that you guys might want to think about going into. So at the macro scale, you see it. At the micro scale, you also see this sort of this funneling sort of thing that Ernst talked about. You see the tracer that is marking these flow pathways. And a lot of it's surely being dictated where those collector channels are, right? You're going to be going to where the exit points are. But a lot of these studies are still yet to be done. But what you have is you've got this pressure gradient coming across the tissue, and Ernst talked about this. It's a cool design tissue where you've got this filter that's presented in front of this resistor. And you've got inside Schlemm's canal, if you have the interocular pressure equal to episcleral venous pressure, you've got red cells that are in there. And this is part of the blood aqueous barrier, right? So this is you don't want these guys and other things to come inside the eye. So this is where the tissue just folds on itself. And so it's very compact when you have this situation. If you have a situation where IOP increases, then you get these giant vacuoles, you get tension that's created in the tissue, you get cells that are stretched, you got all these sorts of things that are going on, signaling molecules going crazy, fluid flow that's increasing. So a lot of things start to happening when the pressure increases. And of course, IOP is generally above EVP most times. So you have these short-term adjustments, but then these long-term set points. And Ernst also mentioned this intricate network that exists between the ciliary muscle and the interwall Schlemm's canal and the JCT TM cells, where you have these elastic fibers that extend up into the tissue and they literally adhere right onto the basement membrane below Schlemm's canal. And this was a stain that Mike Fausch did, and at a relatively modest pressure, this tissue is pretty compact. And then when you get a higher pressure, you see this tissue expand. This is in a human eye. And it turns out this expansion is exactly twice its length, which is what elastin can tolerate, and that's what it allows. And that makes sense because this is a complex elastin structure. And then, of course, the pharmacology is what I love. So you can, we're looking at facility of outflow over time. And then again, we're hitting it with pilocarpine, this cholinergic drug. And we can see if we tug on this network by the ciliary muscle, we go ahead and increase the facility of outflow. You've expanded this tissue and allow fluid to come through here artificially without an IOP increase. And the other thing that Ernst touched upon is this, and Arthur as well, is the dynamics of the pressure. When you go into your ophthalmologist or optometrist and get your pressure measured, it's on one of these points during the day, and it's usually not representative of the pressure. These are monkeys that have been implanted. This was done at Crawford Downs lab. And you can see that, this is when you wake up in the morning, you can see this huge IOP spike that disappears over time, and then, that it changes over the time of the day. And if you look at a shorter scale, so this time frame's over a 24-hour time point, this was a 1 second. You can see these oscillations that are coincident with things like blinking and your heartbeat and other things. So all of this stuff is being felt by the tissues inside of the eye. And that elastin structure that the conventional outflow tract has is built so that it can act like a shock absorber to that, so that it protects the nerves in the back of the optic nerve in particular, from those damaging waves. And it turns out that on top of an average IOP, 15% of the energy in the eye is due to these spikes. And so you can imagine if you have a dysfunctional TM like you would in ocular hypertension, those spikes are going to be much greater because you're not absorbing that energy. And this is just showing the ability of the tissue of the conventional outflow tract. This is a model that I'll describe in a little bit, where you have an experiment where you get a stable baseline and measuring pressure here. It's pressure inside of a chamber of a half of a front of the eye that's clamped into this chamber. So you're measuring the pressure and you're doubling the flow. So you get sort of a steady pressure, and this doesn't have a back pressure, so there's no episcleral venous pressure. So this is minus the 8 or 9 millimeters of mercury that you might have. And then you all of a sudden, you double the flow, you're going to increase the pressure, right? I mean, that makes sense. So you increase the pressure, but look what happens over time. And this is just keeping it at this flow. So at this flow, the pressure comes back to normal, or near normal, or at least during the time of this experiment. And so there's a lot of stuff that's going on here. I mean, they looked at gelatinase A and saw that it went up coincident with that. But there's a lot of other things that are going on so the point is there's a homeostatic adjustment that's going in the eye. That tissue has the ability to change to changing pressures. And this is showing it in another schematic. So how does this happen? So you have low IOP, high IOP. The tissue is deformed and stretched. You have an increase in shear stress that's going on inside of Schlemm's canal as the inner wall moves towards the outer wall. So you have a situation where you have, in all of our eyes, one thing I forgot to mention is that inflow stays pretty much the same. That's not changing. So it's a constant flow system. You have a resistance which gives you an IOP. You have these things that are going on, the shear and the stretch, and so you need to have some sort of response. The gelatinase A was one thing that Ted Acott showed. So you need this feedback system so you can normalize the IOP. And so if you stretch TM cells, for example, this was a review that Daryl Udon did, almost 10 years ago now. You've got all these different things, including gelatinase A, that are going up. So it's not just one thing. You've got all these signaling molecules. And if you look at the shear stress that's in the canal predicted, it's very close to large arteries when you get a full collapse of the canal. So you're getting shear stress that's very similar to you have in large arteries, and has already been mentioned that nitric oxide is one of those things that is produced. And also, you get this alignment around the collector channel. So the cells align with the shear stress, and you can also do this experiment in culture where you have no shear stress, and then you have shear stress, and you can see the Schlemm's canal cells will align, and they'll increase nitric oxide. So the system is very much sensitive to that pressure change. And there's a ton of other things. Nitric oxide is just one of the molecules. You've got all these local mediators. This was a review that Ted and I did about 5 years ago, and it's already outdated. There's a ton other, including just one that we've looked at recently, VEGF, that's also produced as a local mediator. But look at what they have, the impact on facility. They can go up or down. You have a lot of these things that are in balance. So the next part, what I want to talk about is so you've got this system, but really what we care about, over the last 30 plus years, we know some of the molecules that are involved, we know the receptors that are involved, but really what we care about is trying to find a drug to target this tissue, because right now the best we got is what Arthur described, and is either is bypassing it with a shunt or a tube or a trabeculectomy, making a hole in it. So I think that's unacceptable. I think we need to, this is a diseased tissue that's going bad in glaucoma. We need to find a way to target it therapeutically. And so I'm going to emphasize some points that Ernst said. Altering the contractility or the stiffness of outflow cells, we know that this is a component to outflow, and I'll show some more examples of that. Alter the extracellular matrix content. We don't know a lot about the specific molecules and how that happens. That's an area to research. But it also feeds back, and if you alter the ECM, you're also affecting the stiffness. So it's really a yin and yang thing. Altering the permeability of SC, of Schlemm's canal, the inner wall. You need to alter those pores, and we'll talk about that briefly. And then alter the distal resistance. We haven't heard anything about that yet, and I think that that's an important area to think about. So the one thing, this was work that was done with Enhua Zhao and Darren Cheng, who I think is in the audience, at Harvard, looking at the contractility of Schlemm's canal cells, and also we also looked at TM cells. And what you can see in the presence of something that's going to contract the cells or relax the cells, you've got this dynamic range. I mean, it's just crazy the range that these things can stand. And given the impact of IOP on that tissue, you see these giant vacuoles being formed. You can imagine that that's not unreasonable to have that. In the TM, if you hit them with nitric oxide or endothelin-1, also relaxant, a contractile agent, you see that the range that the TM cells are experiencing are greater than a professional contractile cell, like a smooth muscle cell. So it's quite striking how the ability of these cells to deal with these sorts of situations. And if you look at the literature, and you look at drugs that we know that alter the contractility or the stiffness of the cell, all of them that decrease outflow resistance, they have a decreased cell stiffness as well, and vice versa. If they increase cell stiffness, they're going to increase resistance. So in all those cases, it was true. So it makes sense that's an important contributor to outflow. The second one is altering extracellular matrix content. Ernst shows some beautiful pictures of the TM just being loaded with this ECM. I think the most elegant experiment was done in Tate Akot's lab. Here he's monitoring outflow facility. And then these are anterior segment perfusions. So this is, again, a half of an eye that is clamped into a chamber, and then it's perfused. And so the outflow system is isolated in the experiment. And I'll show that in a second. And you can basically see that if he injects in, oops, sorry. Thought I had an arrow, I guess I don't. If he injects in, it's actually a mixture of matrix metalloproteinases. You can see this elevation in outflow facility, but it didn't happen immediately. It took time, and that makes sense because you have to degrade the ECM that's in the eye. And you get basically a point where, after 3 days or so, you establish this new baseline. So clearly altering the ECM content is going to affect facility. And you can do the opposite. And this was an experiment that was done, I believe, when Ross Ether was on sabbatical. And again, Ernst mentioned the importance of TGF-beta2 and a little different system. Well, actually, it was the same system. I believe this was pig eyes. Exchanged it. You have control eyes over time, maintaining the facility. And then the TGF-beta eyes, again, it takes a while for the effect to hit full thing, where you're basically driving the accumulation of the extracellular matrix and getting a decreased outflow facility over time. And if you look at the tissue, you have a lot of alterations in the tissue, in the TGF-beta tissue, including these occlusions and Schlemm's canal and so forth. So the third way is to alter the permeability. Ernst shows some pictures of pores. There are these structures in the inner near giant vacuoles. And you can have basically two types. I favor the border pore. I personally think that the border pore is the active pore, the one that is opening and closing, and that the pore that's associated that we see here is really a safety valve. It keeps the cell from basically blowing off the inner wall. So those are formed when the two membranes, they come so close together that they can fuse and form a pore. And this is showing that same diagram, showing the pores on the inner wall. So how can we change pores, right? So we can learn from the vascular literature, and VEGF obviously is a permeability factor, 50,000 times more potent than histamine. And you think about in the clinic, you've got all these patients that are getting anti-VEGF injections, right? I mean, every couple of months, every month, they're getting an anti-VEGF injection. So they're sopping up, and especially the newer ones are sopping up all this VEGF. Well, so when you measure the change in outflow facility, so these are human patients in the retina clinic at Duke that were being injected with these. And if you look at the number of injections, if you get sort of less than 10, there's a huge spread, and your outflow facility is not really affected. But if you start getting into the 20 to 50 range, you can see that you have a significant lowering of your outflow facility. And so that's suggestive that, maybe VEGF is important in outflow. And if you go ahead and stratify those patients and look at the patients that actually were ocular hypertensive, they had the biggest effect. So these are people that already had sort of a wishy-washy trabecular meshwork, and then you give them anti-VEGF, and you sort of put them over the edge a little bit more than the other patients. And then in the laboratory, Daryl Overby looked at VEGF-A, 164, and saw a dose-dependent increase in outflow facility in a mouse eye. And interestingly, he sort of gets to a point, he gives the thing too much, and then he saw a decrease in facility, which we really can't explain. But clearly, there's an effect of VEGF on outflow facility, and this is sort of where it's at, being pursued further to see exactly what's going on with those eyes. So the fourth way is to alter the distal resistance, and this is all the vessels that are in back of Schlemm's canal. So you've got this circular vessel with these collector channels, and this is all the vessels that's behind that. And it turns out there were some early studies done, actually by 3 groups, but granted the earliest, where they did trabeculotomy. They removed the trabecular meshwork, so the outer wall of the Schlemm's canal was still present, and then looked at outflow resistance, and looked at both normal and glaucoma eyes. And what you see is that after you've post-trabeculotomy, you've decreased the outflow resistance, right? You've taken out the TM. But what was interesting is you still have 25% left. So you still have 25% of the resistance left. And if you do the same thing in glaucoma eyes, most of that resistance that you took out was in the TM, so that suggested that in glaucoma that the TM is important in the resistor, but you can also notice that you have a bit more left in the distal part of the pathway as well. And then this estimate of 25% was actually a bit low. The 2 other studies, Rosenquist and Schumann, showed that it was actually maybe closer to 40 or 50% of resistance. And all of us in the field sort of ignore this. So this is, I think it's important. So what are the evidence that there are contractile elements in this distal area? So you basically want to have these smooth muscle that was shown by Ernst around these endothelial tubes. There was early work done by Annalise Decatur showing that the collector channels actually might be a bit of a sphincter. They have smooth muscle staining around the collector channels. Recent work in mice by Daryl Overby and Elke Lujan-Dreikal showed that there's innervation and there's smooth muscle cells in the intrascleral vessels, so in these vessels that are inside of the sclera before it gets to the episclera. So you've basically got all the components that you need to contract and to affect outflow resistance. Okay, so are there any questions about the 4 ways to alter outflow resistance? Okay, still got you? Good. So what I want to move to is conventional outflow drugs and where they are. So it turns out that we've got 2 drugs that are in late-stage development. They're literally waiting from the FDA any day now to find out exactly whether they're going to get approved or not. All the phase 3 trials look pretty darn good, so there's a good chance that they will. One is a Rho kinase inhibitor. So this is something that's affecting the contractility of the cell, that's relaxing cells, so it makes sense given what we just talked about. And the other one's a nitric oxide donor, which also makes sense given what we talked about. Turns out that this drug is hooked up to latanoprost, and I'll show you how that works in a second. And then there's a few other ones. The rocklatan is also latanoprost plus the Rho kinase inhibitor. And there's a Rho kinase inhibitor improved in Japan, and then also nitric oxide donating, I believe it's timolol, that's also in Japan. So the phase trials, like I said, with Rhopressa look pretty good. This is comparing to latanoprost, so to the first line versus rocklatan, which is the combo drug. And so the way this works is they get patients that are treated, they wash them out, so this is where they are before they start the study, and this is their 8 a.m., 10 a.m., 4 p.m. curve, their IOP curve in those patients, and then you start treatment, and they're going to be on one of those 3 treatments. And what you can see, obviously, is that they all are pretty darn good at lowering IOP. The prostaglandins, we've known that for quite some time. But what you also can see is that there's 2 preparations of the Ropressa, which is a rho kinase inhibitor, and latanoprost are pretty equivalent going through. Then you put the 2 together and you get this additivity, which has really sort of been the holy grail in the field, right? You want to have something that you can get the IOP lower to what we already have on the market. And so this is showing that. This is 3-month data, I believe, and this has been shown out to 12 months, the same sort of stuff. Work that we did with Haiyan Gong, we perfused eyes and we measured outflow facility. Within 30 minutes, we see a significant increase in outflow facility with the rho kinase inhibitors. We measured out to 3 hours. These were in human whole eyes. We fixed and sent the eyes to Haiyan, and she took a look at the histology. And without going through a bunch of pictures, what she saw is exactly what Ernst suggested. If you relax those cells in the JCT, that you're gonna affect that funneling situation and increase outflow, and that's exactly what happened. So this JCT region, the cells, the thickness increased, they were relaxed, and more fluid was able to flow through. So it's exactly as was predicted, which is always nice. And then this is the latanoprostine budone trial, which is this latanoprost plus a nitric oxide donor. And so this is the drug here, and then it has a moiety that it's cleaved by corneal esterases, and then the NO has basically its own half-life where it's released from this arm here. This is the trial results looking at diurnal IOP. This is mean diurnal IOP. And they're looking at 3 concentrations of their drug. And again, remember, this is equimolar with the latanoprost because they're together versus latanoprost itself. Oh, I'm sorry, that looks like there's 4 concentrations of the drug. But the caveat here, and the argument that they make in this paper, is that this is the percent latanoprost that's been shown to give your maximal effect. So anything above that is, you're not going to get any more fluid through the uveoscleral pathway, is their argument. And so, what you're seeing here is a dose response as you increase to a certain level, the amount of nitric oxide that's in the system. So you see an additivity. It's a little over 1 millimeter of mercury. So I think we got it maybe 2 with the addition of the rho kinase inhibitors. So anybody have any ideas why we're not getting more, right? I mean, you would expect to get more than a couple millimeters of mercury additivity to a drug that has a different mechanism. Any ideas? Well, one of the reasons is that latanoprost also affects trabecular outflow. And we never talk about that, but in fact, so there's some overlap in the mechanism of action. And this was studies that Ernst and Elke did early on, showing that also the extracellular matrix and the spaces in the trabecular meshwork are more open. And we and others like Brubaker and patients showed that the outflow facility was increased in those patients as well. And nitric oxide, if you just take the NO, a pure NO donor, you can see that you decrease IOP both in narrow angle and open angle glaucoma patients. And that this was work done with a student that Daryl and I shared, showing an increase in outflow facility with the nitric oxide donor. So it's also, it's decreasing IOP lower and doing that by increasing outflow. So why do we need a conventional outflow drug? So the majority of the outflow goes through this pathway, right? So 70, 90% of it. So there's the potential of having a greater IOP lowering than you would. You want to avoid the surgeries that Arthur talked about. We showed that it was additive, maybe not as good. Maybe it takes a little bit longer to get a bigger bigger delta there. I think the most important thing is you've got a sick tissue, right? This is the tissue that's diseased. And then what do you do? Then you give it aqueous suppressants, then you give it prostaglandins. So you're choking off the nutrients and you're bypassing fluid from it and making it worse. So I really think that if you add a trabecular drug on top of this, what you're going to do is you're going to help stave off some of these disease processes in the tissue, and potentially you might even stimulate some. We haven't talked about it today, but there's a stem cell population in what's called the insert region in the trabecular meshwork. It's out towards the cornea. And there's stem cells there that can be activated and populate the tissue. And then I mentioned briefly that to dampen the IOP fluctuations. Again, if the tissue's functional, might be able to dampen the IOP fluctuations. So the take-homes from this first part are glaucoma is a treatable disease. I was hoping that you get just a sense of the Goldman equation and the relationship to facility and IOP. Wanted to talk about the physiology and pharmacology of aqueous humor production, a little primer of that, the composition of aqueous humor, the physiology and pharmacology of the unconventional route. The conventional outflow pathway is the primary route for outflow. This is the diseased tissue. Self-cleaning filter before a resistor. The inner wall is a one-way valve and is part of the blood aqueous barrier. Outflow tissues are tethered to the ciliary muscle via this elastic network. IOP outflow resistance is dynamic and it's homeostatically regulated by a ton of these endogenous mediators. Conventional outflow is segmental, and we don't quite understand why that is. That we've got 4 ways that we can affect outflow resistance, at least those are the 4 we know now. And that we've got these 2 drugs that are in development. Okay. So let's, I'll just briefly talk about what we have to study conventional outflow. So it turns out culturing TM cells are pretty easy. The TM is this tissue here that you can get. So this is an eye that's on its back, so the cornea epithelium side down. And so you have the cornea here and the scleral here and the scleral spur here. And this is this pigmented tissue here. And it's pretty easy to isolate the TM tissue and to culture cells and get cell monolayers in culture that you can study. And a lot of those endogenous mediators that I described were all discovered with the cell culture. So it's a very robust system. You can get it from human donors, so these are human cells, et cetera. Interestingly, it's a weird cell type, as we said. It has endothelial phenotype, where in this uveal corneoscleral region, and a macrophage-like. So it maintains these passageways, forwards these monolayers, neutralizes oxygen species, et cetera, from the anterior chamber. It's a phagocyte, professional phagocyte. It's fibroblastic in its JCT region, and smooth muscle-like. It's contractile. It turns over ECM, mechanotransduction. And so when you culture these cells, you see sort of this classic endothelial cobblestoney thing. When you get more uveal corneoscleral cells, when you get closer to the JCT, they're a little more mixed, and they tend to be a little more elongated. They still form monolayers, but they tend to overlap their processes a little bit. They like touching, the TM cells like touching each other. And then also, just to make you aware, there's a bunch of transformed cells out there. You need to be aware of the advantages and disadvantages. They're easy to transfect, for example, but they don't express some of the proteins that we know as TM proteins, like mycilin, for example. This is GMT3. This is a review that Abe and I did. This is a transform line. These cells aren't contact inhibited, whereas this is a primary cell line. So you can see the phenotypic differences between the cells. But they each have their role in when you're doing your experiments. SC is another weird cell. It's part lymphatic. It has lymphatic markers and it also has endothelial markers. We've developed methods to isolate them. You put a suture into the canal. It's real easy to feed a suture into the Schlemm's canal. And when you start isolating TM cells, that's sort of the easiest way to get going so that you can actually see the tissue. The TM will be in front of that. And then you let the cells grow onto that suture, and then you remove it later on. Endothelial cells don't like bare, the suture is covered with collagen, and it doesn't like a bare collagen surface. And you can also do it that way, or else as an alternative, use angular porcine angular aqueous plexus cells. It turns out that endothelial cells have a really robust ABC transporter. So if you treat the cells with puramycin and you're not an endothelial, you die. Whereas the Schlemm's canal survives, so you can do a selection based on that. And these cells are a good model for Schlemm's canal. And then there's a few models I mentioned. You can do a perfusion system and measure outflow facility in the whole eye, so a fresh eye like a pig or a human. It turns out that when you do these preparations, you have no episcleral venous pressure, you have no inflow, you have no uveoscleral outflow, or very minimal. So you're totally isolating functionally, and that's why you can do some great pharmacology. You can do these exchanges where you exchange drug into the eye and then measure the outflow facility. So it's basically a system with a syringe pump, a pressure transducer, and a computer, and you're just maintaining, in this case, it's a constant pressure, and then you're measuring the amount of fluid to keep that pressure. And then the other thing is the argon culture system that I talked about. This is more of a long-term, the whole eye is short-term, 5 hours. This system you can keep going for reasonably like 7 to 10 days. Syringe pump, this is just the anterior part of the eye. All the pigmented tissue you take out, otherwise it's going to be just, depositing a bunch of stuff that's not good for the eye. But again, you're functionally isolating this outflow tract. And since it's avascular, you keep it going for that long. And you can also do things like virus experiments. This is where we put a beta-gal virus. So you can change the genetics of that tissue in culture and then study the effects on outflow. 2 new systems, and I'll stop right here, John. There was one that just, Joseph Sherwood, who's here, and you guys want to talk to him. He developed a system for measuring in a whole mouse eye. This is, again, an acute model. It's called the eye perfusion system. And this is in Simon John's lab, an organ culture system for a mouse eye. And the trick there is they use they get a strip of the outflow tract, and then they clamp it in. So you're perfusing a strip of that tissue. But then that tissue is, again, avascular. You can culture it for quite some time. These are papers that just came out if you want to read more. And I'll leave you with that. Thank you.